Okay, so I welcome you once more to our lesson for today. And as mentioned, we're actually going to be looking at genetics. We'll be looking at genetics, and specifically we'll be looking at introduction to genetics. Introduction to genetics. And in this introduction to genetics, we'll specifically concentrate on concepts and terminologies that are used in genetics. So we're going to concentrate on concepts and terminologies that we're going to encounter throughout this particular part of our course on genetics. So genetics, introduction to genetics, look at the concepts and terminologies as well. So in the preamble, we look at what genetics is. So genetics is simply, it's a branch of study. It's a branch of study which concerns in the study of variation and heredity which exist among organisms. So it's a study of variation and heredity which exist among living organisms. So it's a branch of science. And the word genetics simply comes from that word of geneticos, meaning a generative, and genesis simply means beginning. So genetics is a study of heredity and variation. So what's heredity? What is heredity? So when you talk about heredity, we are simply talking about transmission of traits from parent to offspring. We're talking about transmission of traits from parent to offspring. That's heredity. It's concerning transmission of traits from parents to their offsprings. And then variation, when we talk about variation, we are talking about the differences that exist between organisms. So we are talking about similarities and differences. Similarities and differences. So the tendency of the offspring to vary from their parents. The tendency of the offsprings to vary from their parents are what is known as the variation. So that's what we are looking at when you're talking about variation. We're talking about any variance that is existing between the offsprings and the parents from which they came from. That's variation. So if we can see on the pictures there, you can see some variations, some similarities, isn't it? We have
Yes.
Yeah, we have the father, we have the son. You can see that they are exhibiting some differences and they exhibit some similarities, similar with the picture on the right. So this is what genetics is all about.
Sir.
Yes?
Sir. So what you mean is variation. These are the similarities or differences which occur between the parent and the offspring?
Yes. So it's a tendency of the offspring to vary from their parents. So when you're talking about variations, we are going to be looking at, are there any similarities and are there any differences between the offspring and the parent? Is that clear?
Yes, sir.
Okay. So a little bit of some history. We have a man called Gregor Mendel, who is considered to be the father of modern genetics. He's considered the father of modern genetics, because the study of genetics started with the work of this man, Gregor Johann Mendel. And we see that in our modern branches of genetics, there are actually a lot of branches of genetics. We have cytogenetics, we have molecular genetics, behavioral genetics. We have all these sorts of branches. We have all these sorts of branches of genetics. And we see that the father of genetics is Gregor Mendel, who was actually an Austrian monk. Was an Austrian monk who did studies on garden pea. He did his genetic studies on garden pea using 7 characteristics, and we're going to explore more of these in our next session when we'll be looking at classical Mendelian genetics. So in his work, he published what he did, the results, in the Natural History Society of Bern. That's where he published his results. So that's Gregor Mendel. And his work or his publication was known as Experiments in Plant Hybridization, Experiments in Plant Hybridization. And we see that his work was actually recognized for over 30 years. And in the 1900s, we see 3 scientists that were working independently and rediscovered Mendel's work. You see these 3 scientists. We have Karl Correns. From Germany. We have Hugo de Vries from Holland, and we have Erich von Schirmack from Austria. These rediscovered the works of Mendel. So that is just a brief history of genetics. So these findings that Mendel's did are known as Mendelian laws of inheritance. The Mendelian laws of inheritance. And these are some of the things that we're going to be looking at as we continue with our course in genetics. So genetics is very significant, and the works of Mendel's or the works of genetics have contributed tremendously to the scientific world in various ways. Has contributed greatly to the branch of science. So has impacted various areas, such as in the area of medicine, in the area of agriculture, forestry, fisheries, in the region of religion and law. It has impacted all these different types of areas or different types of fields of studies. So it's very significant and important. Other works that we find are the works of William Bateson, who coined the word genetics. He coined the word genetics in 1905. And then the term gene was coined by Johansson in 1909. And we know that the gene is actually the heredity or the hereditary determining factor. The gene is a hereditary determining factor and consists of a continuous segment of DNA. We know that a gene is a segment of DNA. And so through the processes of meiosis and sexual reproduction, we see that genes are able to be transmitted from one generation to the other, generation to the next generation, from the parent to the offspring. And then we see that also the environment has an effect on gene expression. The environment has an effect on gene expression. It plays a role. So it's an issue of nature versus nurture, nature versus nature. So the expression of certain genes is influenced by the environment in which they are expressed. So depending on the environment that an organism develops in, a person develops in, we see that the environment is somehow going to influence the expression of certain genes. So they can either enhance or they can inhibit those genes. So for example, the diet, how someone or what someone eats, and their general well-being, their general health of someone is able to influence even the height, for example, in persons. So different growing environments who can yield a different phenotypical expressions. So studies have been done in identical twins that have shown that even though twins carry the same gene, their phenotypes can be different if they are raised in different environments. So we have examples, for example, height, as I mentioned. The other example that we can give is actually intelligence. Intelligence is greatly influenced by the environment in which someone grows in. So no wonder there is this concept of nature versus nurture, genetics versus the environment, the influence of the environment in which someone grows in. And then we did a look at these things such as the chromosome being an organized package of DNA found in the nucleus of the cell, and largely made of proteins and DNA, which is organized into the genes. We did look at these. Now, when we look at the gene in eukaryotic cells, we see that the gene is going to occupy a specific position on the chromosome. That position is what is known as the locus, or the loci in plural. So locus is a single point. When it is plural, is loci. So terminologies in genetics. Let's look at an allele, or an also known as allelomorphs. These, as we discussed in our other session, we said that these are alternative forms of a gene, and they occupy an identical loci on homologous chromosome. So we did talk about homologous chromosome. We did talk about the allele in our lesson on the theory of inheritance. So when we talk about an allele or allelomorph, simply alternative forms of a gene which occupy identical loci on homologous chromosomes. These control contrasting characters of the same trait. So they're going to control the same trait. So in the example of height, for example, height, an example of height, it's either a character will be tall or will be dwarf. The character will either be tall or will be dwarf. Or we have an allele for purple flowers, and we have an allele for white flowers. And we see that these are existing on an identical loci on homologous chromosomes. So they control contrasting characters of the same trait. For example, we have height. So in the example of the pear plants that Mendel used, we see that we have the gene for height, which has two alleles. It's either it's tall or it's short. So tallness and shortness are examples of these contrasting forms, or the alleles. So they exist in two forms. It's either a dominant allele. It's a dominant allele, or it's a recessive allele. Yes, Eric?
Yes, sir. Sorry for taking you a bit behind. I want to ask a question.
Yes.
Is there a yes, is there a variation in the asexual reproduction organisms?
Is there a variation in asexual reproduction? Asexual reproduction. Okay, thank you for the question. Those that attended
Yes.
our previous class, what's the answer to this question? Guys, is there variation in asexual reproduction? What's the answer to this question if you attended our previous two classes? So there's no variation that occurs, and there's no variation under normal circumstances. There cannot be any variation that can occur in asexual reproduction unless in special circumstances where that genetic variation is introduced. In the case where there's a random mutation that occurs in a DNA's organism, so that can be passed on to the offspring. However, under normal circumstances, there's no variation in asexual reproduction. Remember, meiosis
Okay. Thank you very
brings
much.
about,
Thank you.
yes, remember meiosis brings about genetic variation, brings about genetic variation. Yeah, okay, lovely. So we're talking about the dominant allele. Now the dominant allele is going to express, itself always. So whether it's in the presence of what is known as a recessive allele, the dominant allele is going to express itself. So it always expresses. So it masks or suppresses the expression of the recessive allele. And when we are writing and doing crosses or in genetics, it's the dominant allele is given the letter T, or rather the letter T in the example of height, capital T. The example of height is given the capital T. So the dominant allele is given or symbolized by capital letters, while the recessive allele is symbolized by small letters. So if the dominant allele always expresses and masks or suppresses expression of the recessive allele, it means that the recessive allele can only express itself in the absence of the dominant allele. So the recessive allele expresses only in absence of the dominant allele and is symbolized by small letters, such as the T. The example of tall and dwarf, example of height. So that's a dominant allele and recessive allele. Let's look at genotype and phenotype. Genotype and phenotype. So when we look at the genotype, so from the word gene, remember I looked at the gene. So from the word gene, we have genotype. Genotype. So when we talk about the genotype, we're talking about the genetic makeup of that organism, the genetic makeup of that organism. That's what is known as genotype. And then when we look at the phenotype, we see that the phenotype is the expression of the genotype. The phenotype is the expression of the genotype. So it's a physical features of that organism, the appearance of an organism. So for example, yeah, so I mentioned that it's expression of the genotype. The phenotype is produced not only by the genotype, as we have said, but can also be influenced by the interaction between the genotype and the environmental factors. And I labored to explain on this. So for example, when we are looking at the trait of height, so we are going to have an allele for tall and an allele for dwarf or short. And then we are going to have the genotype, which is T, for example, TT is dominant, or homozygous dominant. And then we have recessive, homozygous recessive TT, or we can have the combination, which is heterozygous. So this will be the genotype. This is going to be the genotype. While the morphological appearance of the organism as being tall and as being short is what is known as the phenotype. So when we look at the phenotype, when we are comparing and contrasting about the phenotype, or when we are contrasting differences between the phenotype and the genotype, we see that we've already mentioned that for the phenotype, it's a morphological appearance that an organism has. While when we look at the genotype, it's actually the genetic makeup of that organism. And we see that the genotype is going to influence or determine the phenotype of the organism and not vice versa. And so the genotype is going to actually remain the same throughout the organism. It's going to remain the same throughout the organism. And we have seen that for the phenotype is also being able to be influenced by the environment in which someone grows in. So individuals usually having different phenotypes, individuals having different phenotypes have different genotype. Well, we know that even individuals that have different genotypes, these may actually have a similar phenotype. So that's about genotype and phenotype. Genotype is the genetic makeup. Phenotype is the physical appearance of the organism. So homozygous, we see that a homozygous is a combination or a condition in which both of those alleles in the homologous chromosomes are identical. So like I explained, like the alleles being either dominant and or being recessive. So in the case of dominant homozygous, we're going to have both capital letters. So in the case of height, we are going to have capital letters T, or in the case of recessive alleles, we are going to have small letters T, small letters T. So that's a homozygous. And then we have heterozygous, in which the alleles on the homozygous, or rather homologous chromosomes, are not identical. So one is going to be dominant and one is going to be recessive. So we are going to have, for example, a capital T and a small letter T in the example of a heterozygous toe, heterozygous toe. So that's a heterozygous and homozygous. And then we have hemizygous. It's a condition when the genes are present only in one copy of the chromosome. So for example, genes on X chromosome and on Y chromosome in males, we see that we have one X and one Y chromosome. And then we have the concept of dominance, simply an ability of an allele to be able to express itself phenotypically, both in the homozygous and heterozygous conditions. And we've talked about this, talking about the dominant alleles. So the concept is simply the same. And then we have the concept that comes into existence as the codominance. Codominance. So what are codominant alleles? So these are alleles that are both expressed in the heterozygous individual. They are both expressed in the heterozygous individual. So they are contrasting alleles. And so in the F1 generation, which we are going to look at, after the parents have been crossed, they are both expressed. So the heterozygote with such alleles is going to exhibit the relevant characteristics of both parents under codominance. The heterozygote is going to exhibit the relevant characteristics of both those parents. So for example, if you are looking at keto, which can be, for example, controlled by two alleles of red and white, we see that if we have capital R's, both capital R's, these are going to signify a red coat. Well, if we have both being W's for white, are going to have a white coat. And then when we have RW, we're going to have a white and a red coat. So we see that the heterozygote has expressed both relevant characteristics of these parents. When we talk about incomplete dominant alleles or incomplete dominance, we see that a heterozygote with incompletely dominant alleles will exhibit an intermediate between the two homozygous parental phenotypical characters. So it's going to get an intermediate. It's going to get an intermediate. So for example, taking an example maybe of a plant where flowers are determined by two alleles, red and white flowers. And when those are crossed, we see that RR, capital R's, are going to signify red flower, while white, or rather WW is going to signify white flowers. So when these two are crossed, if there's incomplete dominance, remember in incomplete dominance, what will be expressed will be an intermediate. So we're going to find that the flowers, instead of being red and white, the flowers are going to be pink. It's going to produce pink flowers. So that's incomplete dominance. It has taken an intermediate between these two homozygous parental phenotypic characters. And then we have what is known as recessiveness. And we've already talked about this concept when we talked about a recessive allele. So this is simply an inability of an allele to manifest its phenotype in heterozygous condition. So in the heterozygous condition, it's unable to manifest. So that's a recessiveness. The other type of alleles that we need to know are known as the lethal alleles or also lethal genes. So lethal genes are simply genes that are going to cause the death of the individual possessing them, either in the prenatal or postnatal period prior to maturity. So you see that the lethal genes usually cause death when they are in the homozygous state. So for example, example of achondroplastic dwarfism. So don't worry about that word. It's just an example. We see that we have the allele A or capital A in a man is a dominant lethal allele in which if an individual has a dominant if an individual has a dominant homozygous dominant, we see that the individual is actually going to die. The individual is going to die before birth. So it's actually aborted. Or when we look at a heterozygous individual, therefore having a capital A and small letter A, those individuals are going to be dwarf. For an individual with homozygous recessive, those individuals are going to be normal. So lethal alleles usually cause death in the homozygous state. The lethal alleles cause death of the individual that is carrying these alleles. So these are some of the terminologies that we have here. So the pictures there are simply depicting codominance and incomplete dominance. So in the case of codominance, we see that it gets both characteristics of the two alleles, gets both. And then in incomplete dominance, we see that the heterozygote or the offspring gets an intermediate characteristic of the two. So that's simply what the picture is showing there. What are the other terms that we find in genetics? So we have hybridization and hybrid in general. So hybridization and the hybrid. So when you talk about hybridization, we're simply talking about a process where individuals are genetically crossed. So the process of crossing two genetically different individuals is what is known as hybridization. So for example, we have a homozygous tall individual and a homozygous dwarf individual. And we have the genotype there, dominant, and homozygous dominant, and homozygous recessive. And crossing these we have, for example, the heterozygous tau. This is what is known as hybridization. So it's simply the crossing of two genetically different individuals. And then the progeny of that hybridization is what is known as the hybrid. So the result of hybridization is known as a hybrid. And then we have the concept of monohybrid and dihybrid. So in a mono simply means one, simply means one. So in a monohybrid is simply an organism which is heterozygous with respect to only one pair of allele at a locus under the stud that we are looking at. So under a monohybrid, you're just concentrating on one allele, For example, you're just concentrating on height, in the case of height. So it's either it's tall or it's short. So monohybrid is just one single property that we are looking at in the example of height. Now, when we look at a dihybrid, in a dihybrid, what we are looking at is an organism which is heterozygous with respect to two pairs of allele at two loci under the study. So in a dihybrid, di means two. So you're looking at two characteristics being investigated or being studied. So for example, you can be investigating on the color of seed and the shape of the seed. So it's either the seed could be, one of the properties, the seed could be yellow round seed, or it could be green wrinkled seed. So you are dealing with two properties. You are dealing with the shape and also the color of the seed. So this is known as a dihybrid. And then we have what is known as a monohybrid cross. We have what is known as a monohybrid cross. So in a monohybrid cross, it's simply a cross between two individual organisms which differ from each other with respect to one pair of alleles under study. So it's only one pair of alleles, as we explained earlier. That's a monohybrid cross. We're only concentrating on one trait that you're investigating. However, in a dihybrid cross, it's a cross between two individual organisms which differ from each other with respect to two pairs of alleles under study. So that's a monohybrid cross. We are concentrating on toe and on height, whether it's toe or whether it's dwarf. In a dihybrid cross, we are going to concentrate on two properties. So you have you are crossing a yellow round seed against a green wrinkled seed. So this is known as a dihybrid cross. So this is about hybridization and the hybrid. What more terminologies do we have? So we have what is known as the P, the F1, and the F2 generations to look at. Okay, so we can just log out and log in back so that we can just summarize from here.